All right, I think we should get started as the people joins in. Good morning, good afternoon, good evening, everybody, wherever you are. I am Brajesh Singh. I work at the Hawkesbury Institute for the Environment at the Western Sydney University. Um, my own research focuses on the soil biodiversity and ecosystem functions and how we're going to harness that to increase the agricultural productivity or environmental sustainability. Today, we are meeting to have a panel discussions organized by AMI, the Applied Microbiology International. And the topic of today's panel discussions is about the topic of the discussion is how we're going to harness the microbial communities for the healthy soils and healthy planet. We have a really good list of people here who is going to contribute to today's discussions. That includes Professor Manuel Delgado Becarizo, who is a senior researcher at CSIC in Spain. His research focuses on understanding of the biodiversity and functions of the soil biome in natural agriculture and urban ecosystems, mainly in context of global changes, including climate change. Our next panel member is Professor Nicola Holden, who is a bacteriologist working in a Scotland's rural college in the UK. She leads a food security challenge center and runs different projects on the microbiome and have a lot of portfolio in the UK research in the microbiome and agriculture productivity. Our third panel member is Dr. Eleonora Agidi, who is also at the Western Sydney University as a microbial ecologist and ARC DECRA Fellows. And her research focuses on plant microbial interactions and how we can harness them to improve the restoration's outcome. Our another panelist is Dr. Jacob John Parnell, who is a microbial ecologist by training and is currently working at the FAO's, United Nations FAO's on the Global Soil Partnership. He is coordinating at this moment the International Network of Soil Biodiversity, also known as NETSO, to implement the Global Soil Biodiversity Observatory. So he will bring a lot of information from the policy sectors, what's going here. I still there is a next one is Dr. Hangwei Hu he's the senior lecturer at the Department School of Agriculture in the University of Melbourne. He is his research focuses in many aspects of microbial ecology, but particularly in the nitrogen's nutrient supply and nitrogen use efficiency. Last but probably the most important panelist is Dr. Sandeepa Kanitkar. She's a founder, chairperson, and managing director of CanBioSys, and she runs a microbial company known for the microbial product in this agricultures. She, starting from her first patent for the producing liquid biofertilizers, which won the United Nations WIPO gold medal, she is involved in improving the efficiency of fertilizers and pesticides using microbes. She is known as a serial entrepreneur and is vice chairman of BSAI, an industry body to the voice, the cause of biological products. Obviously, she will bring a lot of knowledge and discussions from the perspective of industry in this area. So you see that we have a good coverage of different aspects of the science, but also the policy and industry. So we are really looking forward to today's discussions. We will have a series of presentations, short presentations, and after that, we will take as many questions as possible and try to answer as many as questions we will have it. So I will start with my own presentations, if I can find them. So I will start with my own presentations to set the scene. And as I said, after that, there will be a number of short presentations to set the scene for the questions and the panel discussions. I am providing the very brief overview of the role of microbiome in the soil and planetary health. This simple diagrams tells us that why the healthy soil is so critical for the sustainability of our planet. Soils provides every aspects of 95, put in the context, 95% of our calorie intake still comes from the soils. But so the food is the main source of the human's main source still is the soils. But even more, but other aspects is as important. The aspects of the other products that they provide, for example, the medicines, a number of human health for the for the improvement of human health. A lot of medicines is originated from the soils and soil microbiomes, for example, the antibiotics. But there are some harmful microbes in the soil as well, pathogens particularly, and we need to be aware. So every aspect of human health is impacted by the soils where we live. On the other side of that, there's the planetary health. Every aspect of planetary health is impacted by the soil and soil microbiome. For examples, the biomass productions, primary productivity, most of the primary productivity, of course, is done by the plants, but it is a microbial activity that ultimately provides nitrogen, phosphorus, and other nutrients for the plants to grow. So the carbon storage in the ecosystems is a direct result of the microbial activities. And that's mainly through the nutrient cycling. But it also plays a huge role in water cycling, in the purification of water, and the habitats for the biodiversity, create soil habitats where other organisms, for example, protists and the nematodes and other organisms go and live, so have a direct contribution in the planetary health. The importance of the healthy soils for the human society can be gauged from this slides from the Global Soil Partnership. And what you will see that almost all sustainable development goals of United Nations requires healthy soils. So that's how important the healthy soils is for the humankind. And that is important, and because as we talk about how they are important on the crop micro productions, filtration and buffering, climate regulations, which is we haven't talked about, the productions and the mitigations of the greenhouse gas is mainly carried out of the microbes that lives in the soils. And so the healthy soil is important, but that healthy soils, in fact, is a medium that provides the optimum microbial activities, because the soil functions, particularly the processes, are carried out by the microbes, by the soil biota, but mainly with the soil microbes. So the soil microbes is critical for healthy soils and ultimately the functions and services they provide. And why it is important, this is in relatively new informations, that the soil is the home of the most of the global diversity. 59% of the global biodiversity lives inside the soil. And that's why it is so important to provide all the services and functions they provides for they provides. I'll take some of the examples. I briefly said that this is critical for the climate regulations. Why? Soils stores more carbon than the plant and atmosphere together. The soil organic matter, which have around organic and inorganic, if you put together around 2,300 or even more, carbon is stored in the soils, while the plant and atmospheres have a substantially lower. Plant biomass has around 550 petagrams to the atmosphere, around 800 petagrams. So because soil has stored more those carbon in the systems, that's how they regulate. If we lose even 25% of those stored carbon, it will dramatically change the climates that we have not known, and probably our living systems will collapse. That's how they impact the carbon. But the carbon is just one. Methane and nitrous oxide, or almost all methanes are produced, and the biological methane is produced by the microbes, and almost all biological methane oxidations or consumptions is done by the microbes. So they are critical component, critically important for most of the climate regulatory greenhouse gas emission and storage and mitigations. It's also a talk about how important it is in the terms of the soil in the human health. As soil is a home of many pathogens that can directly impact. So, for example, E. coli, Clostridium's number of Clostridium pathogens, the Enterobacter, Campylobacter, E. coli, the Listeria and Salmonella. They all have a life cycles that they survive in the soils, and there is through the direct contact or through the food can infect the human beings. This is just one of the examples of the Aspergillus, which causes around 2.1 million infections every year, and more than 85% of people with immune compromised systems dies of that. And even the healthy populations, 18.5 population, once the infection is there, will die because of the resistance to the antifungal agents. But they are also a source of a significant sort of antibiotic resistance itself. A lot of antibiotic resistance is developed in the soils, and then through the different systems is a migration to clinical settings, and that makes the treatment of infections difficult, and some strains become so resistant that the antibiotics becomes ineffective. That's from the human health point of view. But if you look from the plant point of view, in fact, there has been not an era in the human civilization history that has not seen a rapid or a really catastrophic plant infections that has not caused a significant famine. This is just one of the example, and the figures is the Irish famine in the last century that was caused by the Phytophthora infestans infecting the potato lead blight, so the disease called lead blight. It killed million people, and millions more has to migrate because there was no food in the systems. But it's not only the pathogens. One of the things I will argue with later on, that if your system is more diverse, you have a less pathogen in systems. And this infections, this sort of problem is only going to be the worst in the future. And because a lot of modeling projects that the pathogens, the plant pathogens here, for examples, their abundance and the distributions will expand. As a result, there will be more pressures on agricultural productivity, and that then will compromise the goal of food security across the globe. The second part of that is, but this is a problem. Microbes can create some problem, but it is also solutions of the most of the problem the human society is facing today. For example, the climate change. We talk about how they can play a role in terms of the carbon removal from the atmosphere, as a result, regulate the carbon. But the atmospheric carbon emission is increasing because of the industrializations and human activity that will keep increasing, at least for time being, and that will release into the temperature. So for the continuous food supply and good food systems and food security, we need to have agriculture system that is productive. And if you look at all those sort of literature that's coming up, at least in the short term, and when I'm talking about short term, I'm talking about decades to centuries, the adaptations of those plants, particularly the perennial plants, will come from the microorganism host rather than the plant itself, because the evolutionary response of the plants will take millennia, particularly when you're talking about the perennial plants. And that's then has given the focus on the using on the different tools of microorganisms. And one of the tools have been, and probably have been being used for centuries, honestly. The rhizobium has been used for more than 100 years. to use the microbiome to increase the farm productivity, at the same time reduces the environmental impact of the agrochemicals. But one of the problems have been with the use of microbiome is they are pretty inconsistent in the field. It works in some conditions, doesn't work in other conditions. And I guess there is a number of issues that results into that inconsistent effects, which some of them are the lack of product diversity, for example. We know there are a million species of microbes, even hundreds of millions of species of microbes. But if you look at the product, microbial inoculant product that is in the market, are the Bacillus, Pseudomonas, Streptomyces in biocontrol, and also in the phosphorus mineralization and some of the rhizobia and azotobacters in terms of the nitrogen fixations, trichoderma, maybe some mycorrhizal fungi in the fungi. So the most of the product those products have been developed because they're easy to culture, easy to ferment and then formulate, but there is a real potential that we have not harnessed. And other critical component is the microbes living system. So you have to isolate them, culture them, ferment them, and formulate them, and store them, and transport them. And that has to be the cells that needs to be viable and alive in all these conditions before it goes to the plants. And that is not happening, and that's the result. We have inconsistent effect. So some of the proposals that comes into the discussions, and we probably will discuss today, is that rather than the shotgun approach we have taken for the development of microbial inoculants, we need to targeted products that originates from the crop core microbiomes or pathobiomes, so that they will able to go and colonize, understanding more what microbial able to go and colonize the plants, because this is a critical. Unless microbes able to colonize the plants, you will not get a performance of the plant on the plant productivity or plant health. So we need to have more fundamental understandings in terms of what makes a microbial inoculants more productive, and what makes them what microbial inoculants are more able to colonize, and what we can do to increase the colonizations. And one of the arguments I always argue is that understanding the eco-evolutionary interactions between microbes and microbes, but also in the microbes and plants, will allow us to develop inoculants. Those are more effective in colonization of the plants. I will finish my presentation saying that these sort of issues cannot be solved by the one or two research group or in the university or organizations. And therefore, there's a need to work together in this area from the national NGOs to the United Nations bodies. And this is what we are trying to do, work with the different partners across the globes to deliver on this side of projects. So thank you very much. That will be my introductions of this presentation. And now I will hand over to Manu to give his brief presentations on the topic. Manu.
Perfect. Thank you, Raj. I'm going to share my screen. All right. Can you see my presentation?
Yes.
Okay, perfect. First
you want to put on the presentation mode? Sorry, Manu. Do you want to put it on the presentation mode, if you don't mind?
Okay, let me try.
It's gone in presentation mode now. It's okay. All right, it's presentation mode now.
Okay, perfect. Yeah, thank you. Well, first of all, I would like to thank the organizer for inviting me to be here today. It's really a pleasure. Today, I'm going to talk to you a little bit about conserving soil biodiversity to support soil health under global change. First of all, I would like to pretty much support Brajesh in his presentation about the fundamental importance of the biodiversity to support ecosystem function. In particular, we know we have been studying this for a while, that the biodiversity of keystone species is critical for supporting the biodiversity and the function of the ecosystem, including the productivity of the ecosystem, the decomposition of the organic matter, the nutrient cycling, pathogen regulation, etc., etc. As you know, soil organisms, they interact with each other in complex networks, and within these networks, certain group of taxa, the biodiversity of this taxa is critical to support function. We have seen this in natural ecosystems and also in cropping ecosystem, in agricultural systems. And in fact, many of these species, like for example, Rhizobium, Agrobacterium, Burkholderia, etc., they are commonly used in bioproducts in microbial consortia to support the productivity of the croplands. However, I would also like to stress that soil system, soil health, biodiversity of our soil is highly vulnerable to global change. And of course, climate change, warming, increases in aridity, for example, and also land use intensification, including the conversion from urban ecosystems, from native environment to plantations, urban environment, cropland, grasslands, etc., is very critical for the biodiversity and the function of our systems. And I would like to put you a few examples of this from our own research. Back in 2020, we found, for example, that increases in temperature, both at global surveys and also in long-term experiments, they can have a positive influence on the pathogens, on the plant pathogens that live in our soil. Put simpler, warmer soils, they also have a greater proportion, a greater dominance of pathogens. And pathogens like Fusarium and Alternaria, as you know, they are fundamental for the productivity of the cropland, of the agricultural systems, for example, in wheat fields or for the production of apple, etc., etc. So it looks like a warmer planet will also have more pathogens. Not only that, over the years, we have been conducting a series of studies that have demonstrated that increases in aridity, especially in dryland ecosystems, that they are already dry ecosystems. You need to understand that drylands, they cover almost half of the planet already. Increases in aridity in this type of system can have a very negative influence on the biodiversity of our soil, on the function of our soil. Especially important, increases in aridity at certain level of the aridity gradient, for example, in the transition between semi-arid and arid ecosystem, can have a critical, a drastic negative influence on the productivity of our ecosystems, reducing the amount of carbon that our ecosystem can storage, reducing the biodiversity of the system, increasing the amount of pathogens, etc., etc. So increases in aridity in an already dry wall is going to have an impact on the biodiversity and the function of our soils. Also, more recently this year, we find out that deforestation pretty much changes transformation of native forests to plantations, to grasslands, and to croplands, can have a critical effect on the soil health and also on the biodiversity of our ecosystems. For example, we know that transforming a native forest into a plantation, a grassland, or a cropland can result in important reductions in the carbon of our soil. It can also result in a rejuvenation of the bedrock. So we have the bedrock is coming to the surface, so maybe we have more phosphorus, we have more pH. But also very important, our systems, they become dominated by pathogens instead of by symbiotic organisms, like, for example, mycorrhizal fungi. And also, our system become very, very homogeneous. So we only have the same species of fungi, for example, and this is having a critical impact on our soils. Can you see my presentation now?
You can see it, but it's quite small for some reason. Don't know why.
Okay, but you can still see it, right?
Yeah, I
Perfect.
think that's here. I think this is not presentation mode, but slide is bigger. Maybe just use this way, Manu.
Yeah, I don't know how to put that in layer. I'm not sure exactly how to do that. So I will. Let me see. Maybe. Let me just stop this for a second.
Yeah, it's gone back to the same. It was. I think probably just don't do the. Yeah, this format is okay, man.
Okay, so now you can see it like larger.
Yeah.
Perfect. Yeah. Well, so yeah, as I was just mentioning, the deforestation, for example, as an important land use intensification can have a critical effect on the biodiversity and the function of our soil. And recently, we have been actually investigating how multiple global chain stressors they affect the soil simultaneously. As you know, our soils are subject to multiple global chain stressors simultaneously, not only to one of them. For example, they are subject to increases in aridity, especially in drylands, increases in temperature, seasonality, salinity, the pH changes, heavy metal, human impact, etc., etc. So what we have found is that soil biodiversity is very vulnerable to an increase in an increasing number of stressors. So as soils that they are very stressed out, they can have lower biodiversity. And as you can see in this presentation so far, soil biodiversity is important for function. We also know that this biodiversity and function is highly vulnerable to the global change. And I believe this knowledge is critical for the producing synthetic communities that they make sense for our ecosystems. For example, selecting locally adapted microbes that they can actually support the health of our ecosystem. For instance, in this publication last year in a greenhouse experiment with maize, we found that syncoms, synthetic communities, probiotics, developed with native microbes, they are actually especially important for supporting the function of poor soils. Very interestingly, commercial syncoms, they didn't work very well in our soil because these commercial microorganisms that they normally come from other type of soils, they actually they are not very good establishing and colonizing the soil system. And however, the native microbiome is already adapted to the conditions of our soil. So the native microbiome is very important when supporting production in our systems. Another example of this is the soil, healthy soil, the microbiome of healthy soil that can be used to actually fight pathogens like Fusarium oxyporum. In this study, we found, for example, the native microbiome inoculations from healthy soil that they were not affected by Fusarium. They actually can increase the resistance of soils that are unhealthy, that they are already affected with this pathogen, to suppress root disease. So it looks like native microbiomes, they play a fundamental role compared to, for example, commercial bioproducts. In a sense, we are really understanding now that the contribution of soil biodiversity to support function is very context dependent. And that is what we found a few days ago in this publication, where we found that soils that they are actually very dry, very poor, very young in terms of geologically young, they are very, very dependent on the soil biodiversity. However, older soils, mesic ecosystems, they are not that dependent. And the reason is pretty much the stock of organic matter. In a very dry soil, in a very poor soil, we don't really have a very big stock of organic matter. So the soil really depends on the biodiversity, on this kit of tools that the biodiversity provide to decompose the organic matter, and they really depend on a daily basis on this soil biodiversity. And so far, biodiversity is important, is highly vulnerable to global change, and this adapted microbiome can play a fundamental role for the probiotics. And because of that, it's essential that we learn how to protect, how to conserve the biodiversity and the function of our soil for the future generations. And the first step for that was actually identifying the global hotspots for soil biodiversity and function, for instance, for the number of species of our soil, ecosystem services, how unique our soils are in terms of microbial communities. And that was what we tried to do a couple of years ago in this study, where we identified the global hotspots for soil biodiversity and function. Unfortunately, in this study, we also found that most of the soils that they are considered hotspot of biodiversity, they remain unprotected from the point of view of natural parks, national parks, etc., etc. So it's very important that we learn how to protect the soil, this native soil especially, if we want to leave the future generation with these tools to produce more food in a context of synthetic communities. With that, I would like to leave you with a very simple take-home message. Biodiversity is important for function, however, it is highly vulnerable to global change, and we need to understand much more about how these native microbiomes can help us to produce food in a context of global change. With that, I would like to thank you for your attention, and of course, of the funding agencies. I'll leave it with the next panelists. Thank you.
Sorry, I was muted. And so now I would like to invite Professor Nicola Holden for her presentation. Nicola, please.
Thank you very much, and great to be here. Thank you very much to AMI for organizing such a fantastic session. And yeah, I would like to just spend the next 10 minutes just giving you a brief overview of some of the resources around soil microbiomes that we've got here in the UK. So if I can just share that screen. Okay, hopefully you can see that okay. So yeah, as I say, we're going to look at some of the resources, but before I get there, I just want to tell you a little bit about our journey around microbiomes in the UK and a couple of the sort of seminal milestones. One where we kind of got together as a nice group of microbiologists together with the stakeholders, looking at how we could actually apply our knowledge around microbiomes and you know, get some more attention around it. So that was one of the documents, Unlocking the Microbiome, that was published back in 2017. And then a few years later, we got together with one of our industry-supported funding agencies, and we produced this microbiome strategic roadmap. And I think these are really quite important documents, and they've kind of helped to shape our research around microbiome and, as I say, application. And at the same time, we're now in partnership with some of the global movements. So I've got a couple of examples here, one from the EU, and I'm sure you're very familiar with microbiome support, and also the Phytobiomes initiative out of the USA as well. Okay, so the couple of examples I wanted to show you is the Great Soils Project, that was Soil Biology and Soil Health Partnership. This is now completed, it is a mature project, and I would recommend that you go to the website to have a look to see what was being done there. It was run out of NIAB, and we had lots of different partners involved in it. It was a very large-scale project. And one of the things that we wanted to do was really look about, can we use our knowledge for different soil microbial functions to help develop application information. So really developing scorecards around soil health that some of the primary producers could use. So I've just highlighted a couple of the projects there that are focused on the microbes and the microbiomes, but it's really part of a much bigger project. So as I say, there's a lot of resources already online, and I would recommend that you have a look at that. I've just divided that into sort of the molecular approaches, looking at soil health assessments, for example, or indicators of soil microbial activity, and how cultivation affects the soil microbiology as well. And then some of the management approaches, because what we wanted to do was really make the outputs of this project applicable for the primary producers, so for the farmers in particular. So for just zoning in on one aspect was looking at DNA to assess soil health, and we had some quite well-defined tasks for that, and we could see some of the major drivers around pH, for example, or organic amendments. And we were able to utilize some of our long-term experiments. So we had one that was set up in 1961, which we are still continuing to this day, looking at pH, for example, and we do that up in Aberdeenshire in Scotland, in the northeast of Scotland. We are able to continue this project with Scottish Government funding through an organisation called RESAS, where we're looking at the function, microbial function, under the Healthy Soils project. Another project that is currently on the go is generating a biological and bioinformatics resource around crop microbiomes. And what we want to do is to make this resource available for the agri-tech industry as well as for researchers. There's a lot of organisations involved in this, and it's run by CABI in the UK. And again, I would urge you to have a look at the website to find out a little bit more. And what we've done is we've taken some of the major soil textural classes from across the UK and set these up in a glasshouse experiment where we can grow different crops, and then we can understand what's going on with the microbiota. But one of the keys to this is we are generating a biological resource. So we are storing everything. At CABI, they're cryopreserving the actual microbiomes as well as culturable isolates, whereas I'm involved in the sequencing, for example, and we have colleagues in John Innes Centre who are involved in setting up synthetic communities. So for the sequence-based analysis, we're doing lots of different types of sequencing, whether it's metabarcoding at the amplicon level, shotgun metagenomics, or whole genome sequencing of the cultured isolates. So all of that information is being drawn together into a database, and that allows us to do a cross-comparative type of work. So for example, I've got a partnership, international partnership with Argentina, with the organization INTA, and we can then do comparative work between their systems and what we have in the UK. So this again is a really nice resource that we want the community to be able to use and access. And then finally, I was just going to touch on a different type of environment. Soil just isn't just on the outside, it's also used in the inside. So we were asking questions around controlled environment agriculture and growing crops in these types of environment. What happens to the microbes? I'm interested in pathogens, for example, and does this sort of situation then increase the likelihood of transmission of pathogens? So I was able to answer some of these types of questions, again, using a microbiome approach. And over and above the questions around pathogens, it's actually been really useful to understand where the plant-associated microbes have been coming from. So they're not coming from the water or the irrigation sources. They're really coming from the soil substrates, from the compost that we're using to grow the plants in. So this, again, is producing information that we think will be useful for some of the adopters of this type of novel technology. So yeah, I'll just finish that quiz, a quick whiz round with acknowledgements for some of the major projects. And again, I'd urge you to have a look at the websites if you want to have a look at some more, or please feel free to contact me as well. Thank you very much.
Thank you, Nicola. Thank you very much for your presentations. Can I request all of them to keep the questions coming? We will take these questions and answer sessions once every presentation is finished. So please keep putting your questions in the Q&A, and we will tackle all those questions, as many as possible, after those series of presentations. Our next presenter is Dr. Eleonora Aguirre, and she will talk about the microbe soil health and restorations. Ellie.
Thank you, Ross, and hello, everyone. Okay. So as Russ mentioned, I'm interested really in the use of microbiomes and restoration, and I'm based in Australia, although you cannot tell from my accent, but which is a beautiful country. I really like this quote from Dorothea McKellen, "A land whose weeping plains or rugged mountains, drought and flooding rains," really captures the diversity and the beauty of Australia, where I'm so lucky to live. And Australia is probably one of the largest countries that are dominated by drylands. We call it rangelands, or the outback, that occupy about 80% of the whole landscape. You can see creating beautiful landscape for us to appreciate. But Australia is also one of the countries that has changed the most in the last 200 years after the European settlement, because nowadays you can see how agriculture dominates the landscape. In the left picture here, you see the purple and pink areas are the areas that are really dedicated to nature conservation or protected areas. Everything else is pretty much devoted to agriculture, either cropping or grazing. You see another representation of this impact of human activities on the actual landscape and how invasive it is from spatial point of view. So Australia has changed dramatically in the past 200 years. And these activities have profoundly impacted the occurrence and health of native vegetation. This is a snapshot of New South Wales, where Sydney is based. So you can really see how vegetation pressure, mainly land use change, have threatened most of these communities. You see here how this is the area where in the middle, the really dark green one, dark red one, where Sydney is, so the most intensive land uses, but then you see there are some conservation area in green here. Everything else is, in the yellow area here is agriculture, dryland agriculture and plantation. And this land use change cause habitat loss of ecological functions, encroachment, establishment of exotic species. And you can see it here, how it has changed compared to the pre-European settlements. So everything that is pretty much orange means pretty bad. So means that less than 20% of the land is still preserved compared to the pre-European settlement. So there is a huge pressure to preserve what's left of native vegetation, and to also try to restore and reintroduce some of the native plants that have been lost. Just to give you an idea, in the last 10 years, 2 major areas in New South Wales have been declared threatened as threatened connected communities. There are more than 200 species or communities in only New South Wales that are considered endangered. And those require active conservation and constructions, which comes at a huge cost and represent a major challenge. The main challenge is probably the establishment of the seeds because of weed invasion, overgrazing, but there is continuous disturbance by feral animals, and there is a huge change in the fertility, in the chemistry of the soils that has happened over the years. And this really makes it very difficult to re-establish these native plants or even just protect them. So, and one of the main reasons is that even if you get, I think, the chemistry of the soil right and try to balance and manage the, for example, establishment of invasive plants, what is really difficult to reintroduce and reestablish is the mutualistic connections that have been lost with land use change. And this is the main challenge. For example, for mutualists, one that lost, they need to be actually reintroduced. It's not possible to just change the management practice to reintroduce them. and in particular microbial communities, which is the focus of the work I'm doing. At the moment, we are trialing using a home-based solution that is based on the use of endophytes from native plants for biopriming seeds. So trying to actually re-isolate some of these mutually lost microbes from healthy plants, from healthy communities, and reintroduce them to enhance biopriming the seeds, so adding them to the seeds directly to enhance the germination, viability, and vigor, and hopefully also a biotic stress tolerance and biotic stress tolerance. We are trialing some of these approaches in the Cumberland Plain conservation areas, again in the Sydney Basin region, which is expanding more than 11,000 hectares. It's a highly degraded, and there is we're targeting this area from different point of views. One of them is using microbiome-based solutions. This approach, the approach we're using is at the moment targeting 4 native grasses that form they're keystone, considered keystone grasses in the area, and isolating microbes from those grasses and by prospecting them for plant growth promoting properties. We have some preliminary results, so this is not published yet, so take it with a grain of so it's just preliminary results, but we see that, for example, there are multiple traits present in terms of nitrogen fixation, phosphorus solubilization, and production of phytohormones. There's a lot of these traits that occur in microbes derived from seeds, so it seems like seeds somehow carry these plant growth promoting properties. The current work is on field trials, field trials and optimization of the delivery modes. And that's all from my side. Thank you.
Thank you very much, Ellie, for the very good presentations. Our next presenter is Dr. Hangwei Hu of Melbourne University. Hangwei, please.
Thank you, Brad. I'm going to share my screen. Okay, I hope you can see my screen now. Yeah, first of all, I want to thank the invitation by AMI. Hello, everyone. My name is Hangwei Hu. I'm a soil microbiologist from School of Agriculture, Food, and Ecosystem Sciences from University of Melbourne. My research is mainly focused on biogeochemical nitrogen cycling processes, and I'm particularly interested in using a microbiome-based approach to improve the nitrogen use efficiency. But over the past 10 years of studying nitrogen cycling process, I realized a microbiome-based approach alone cannot solve all the problem, and probably we need a multidisciplinary approach for enhancing the nitrogen use efficiency. This slide is so I got a photo from Brad paper published in Nature Review Microbiology. This picture showing 3 of the key process of nitrogen cycling steps, including a nitrogen fixation, nitrification process, and a denitrification process. So we know in Australian agriculture, over 3.5 million tons of nitrogen fertilizer is used annually, and which can cost about 4.5 billion per year. However, the fertilizer nitrogen use efficiency is very low. For example, after we applied nitrogen fertilizer into the soil in the form of ammonium, ammonium can be converted to nitrate, but the nitrate is easily leached into the groundwater, which have some environmental pollution issues, and also nitrate can be converted to the greenhouse gas, nitrous oxide, which can have some climate change effects. So this nitrogen loss sometimes can account for more than 50% of the applied fertilizer nitrogen into the soil. And also such natural loss represent substantial financial losses to the primary producers, and also it is a significant societal and environmental damage cost, for example, which can cause climate change, biodiversity loss, and environmental pollution. So currently, some of the mitigation strategies include controlled released fertilizers. For example, some nitrogen based fertilizers are control released, which can better match the crop nitrogen demand and the nitrogen supply. But currently, the commercial controlled release fertilizers are quite expensive and are not cost effective. And also, we can use ureas and nitrification inhibitors, but usually these inhibitors have inconsistent performance. For example, this commercial available nitrification inhibitor, DNPP, is quite effective in neutral or alkali soil, but they are not effective in acidic soil. So therefore, we require some novel approaches to address this critical challenge, which is the low nitrogen use efficiency, through some of the cost-effective control, release the nitrogen fertilizers, and new nitrification inhibitors, and innovative microbiome based products. So I'm going to introduce one of the key projects that we are now doing in the University of Melbourne. So 3 years ago, we established a research hub for smart fertilizers. So this hub or project was funded by Australian Research Council. And this project is a partnership between university researchers across different disciplines and the fertilizer industry. So we are working together to transform agriculture by delivering some new nitrogen-based fertilizers and also decision-making tools which can help enhance fertilizer nitrogen use efficiency. So this is our team. We have more than 40 or 50 members. So this hub brings together 5 interconnected and multidisciplinary research teams. For example, the team 1 is led by chemical engineering scientists from Melbourne University. They are working on the fertilizers with some novel engineered coatings. And the team 2 is led by organic chemistry scientists, also from Melbourne University. They try to design some innovative ureas and nitrification inhibitors. For team 3, it's led by myself. We are working on to have a better understanding of the plant microbiome interactions, and particularly we are assessing the new compounds from team 1 and team 2 on the plant and the microbiome interactions. And also, I feel very important is we need to assess the agronomy, environmental, and social benefits of new products, if our products can make any impacts. And also, we have a team 5 is led by economists. So this team trying to provide a framework to drive the commercialization of our new products from team 1 to 3, including both chemical and biological fertilizers, and which can also contribute to the adoption of these products by the producers or by the farmers. So I just have a brief introduction of some of the work in this research hub. For example, the team 1 aim to develop a new class of fertilizers with engineered coatings to enable controlled release of nutrients and also nitrification inhibitors, which can work in a range of soil types, different climate conditions, and the diverse agriculture settings. So which can work for different crops. And importantly, we try to design some new coatings which are low cost, efficient, highly flexible, and environmental friendly. So this is quite important. This is microscope image showing some of the new coating materials. As you can see from this picture, this is the coating polymer-based layer materials. We can see many holes in these materials. Like these holes can allow the release of fertilizer into the soil once we apply this controlled fertilizer into the soil. And also, which is very important, we need to assess whether these products are environmental friendly. So we conduct some soil microcosm incubation to make sure these new compounds or new coatings are biodegradable. For example, we can compare the control treatments and the post-soil incubation treatment, and we can see the changes of the coating's surface and ensure these coating materials can be degraded by soil microbes. And the SIMS tool is working on new ureas and the nitrification inhibitors. Currently, actually, we don't have a lot of commercial available ureas or nitrification inhibitors on the market. For the urease inhibitor, so this inhibitor is working on inhibition of the hydrolysis of urea, which is a nitrogen fertilizers. This urease inhibitor is trying to slow down the conversion of urea to ammonium. So this is the urease inhibitor, but this inhibitor can only last several days, sometimes just less than 1 week in the soil. And DMPP, which is currently commercial available nitrification inhibitors. So this inhibitor can slow down the nitrification process. So by this way can reduce nitrogen losses from the soil, but this commercial available nitrification inhibitor is more effective in alkali or neutral soil. So now in this hub, we're trying to design some new inhibitors and do some structural variations of this commercial available inhibitors. Actually, we have produced more than 10 or 20 different types of new inhibitors. Now we are trying to upscaling and think about how to commercialize these products. And yeah, some of the products are quite effective in acidic soil and can be comparable or even have better performance than the commercial available DMPP. And also they are not toxic to the plants, as you can see from some of the photos from the glasshouse study. And for SIM3, we are trying to have a better understanding of the plant microbiome interactions. And in particularly, we are interested in nitrogen-fixing bacteria. As Elena introduced, we are interested in indigenous or native crop microbiome because for this indigenous plant or called wild relatives plant, they have a long evolved with the soil microbiome. So usually the wild relatives are associated with a higher diversity of soil microbiome, and they have more plant beneficial microbes, and usually they have a higher nitrogen use efficiency. So we try to isolate some of the new nitrogen fixing bacteria from the indigenous plants, because Australia is a plant, it's a continent for many indigenous plants. And also we propose some pipeline for combining Raman spectrophotometer and with stable isoprobing techniques to better isolate the nitrogen fixing bacteria from the wild relatives, and they followed by some synchrone testing on the control the conditions and also in the field conditions. And also for myself, I'm very interested in biological-based nitrification inhibitors. As we know, soil virus or bacterial phages, they can control and also they can kill nitrifiers. So I'm interested in the isolation of some bacterial phages from soil or from different environments, and try to identify some bacterial phages which can exclusively work on the nitrifiers, for example, ammonia causing bacteria and archaea, so that we can use bacteriophages based tools to better control the or slow down the nitrification process, so we can contribute to the enhancement of nitrogen use efficiency. So that's all from me. Thank you.
Thank you very much, Angwe. Appreciate it. Our next presenter is Dr. Jacob John Parnell from FAO. Jacob?
Thank you. Can you hear me? Do you hear me?
Yes.
Okay.
Yes.
Let's see what's going on with my presentation here.
There it is.
Okay. Can you see that?
Yes.
So thank you, and I have very much enjoyed the topics and the themes, the science that has been shown so far. I'm going to talk a little bit about some of these concepts as well from a little bit of a different perspective of kind of a little bit of kind of the policy and some of these other kind of avenues. Brajesh nicely pointed out how many of these ecosystem processes and ecosystem services we rely on in the soils. He pointed out beautifully that all 17 of all 17 of what we're looking for as far as our goals, sustainability goals, are tied to soils in some form or another. And one of the problems that we have, and Eleonora described this a little bit in the kind of a more regional approach, but we know that about 33% of the world's soils are currently degraded. And if we don't try and do something about that now, then by the year 2050, the trend is that over 90% of the soils could be degraded. And one of the issues that I find is that most of this degradation has been based on other measurements. So this is not looking at biology. This is mostly looking at physical and chemical parameters of soils to show that they are degraded. And so in a way, we don't really know what this looks like as far as biology is concerned, that 1/3 of the world's soils are degraded. So Brajesh and also Manuel pointed out that soil biodiversity could be a solution to a lot of these global problems. I've got a lot of those listed out here. But it's also alarming that we see that the more stressors that we put on soils, that impacts the biodiversity. And of course, the biodiversity is kind of the buffer for those stressors. And so at some point, as we lose biodiversity by putting more and more stress, we come to a catastrophic failure of a system where we start to really lose ecosystem services. And so part of the question is, when do we hit that? Do we hit that? What does that look like? And so because of that, there are a lot of policy groups, there are governments that are starting to take notice of this. So in 2022, at the Convention on Biological Diversity, the Convention of the Parties, one of the main components that I am, of course, very excited about was they really started to take note of soil biodiversity. And there are two outcomes of this that I think are very important. One is the recommendation that the Food and Agriculture Organization implement or start kind of putting together an international network on soil biodiversity. So this is going to be scientists, it'll be policymakers, it's farmers, practitioners, it's anybody that's interested and wants to be involved with soil biodiversity. Now, the second part of this is to implement a global soil biodiversity observatory. And so the point of this is we see the biodiversity or soil degradation is happening. Eleanor also showed that the ecosystems are degrading. And so what does that look like below ground? So what is happening below ground to our soils? And we haven't measured that, at least not at a global scale. And part of that is, as Nicola pointed out, is that we're not always measuring the same things. And so we need to have some kind of harmonized, some kind of a standard of how are we going to measure soil biodiversity so that we can actually compare different sites, different areas, and that we can then compare and look at what different treatments, what different practices are going to do to hopefully restore some of that soil biodiversity or at least protect it. And particularly, as Manuel also pointed out, in areas where soil biodiversity is most threatened. And so, let's see. So if you are interested in joining this international network on soil biodiversity, I would recommend, I think you can scan this QR code and then you can click on this tab that says, how can I join the network? And it'll pop up a form where you'll be able to fill out your information and then you can be part of this network. And one of the main goals of this network right now, so this network will continue, but right now, one of the main goals of this network is to really establish a global soil biodiversity observatory. And we're just barely in this implementation phase. So this is the phase that is being driven by the Global Soil Partnership and really the board of this international network on soil biodiversity. And the first step of this is really to define what should we be measuring. It would be great if we could do sequencing everywhere, but there are some countries that don't have the capacities to do sequencing, and so we need to start somewhere. And so we've got we've defined 3 tiers of biodiversity variables. And so one tier is going to be something that any lab can start measuring within a couple of months, so that network members or the GSP can basically build that capacity within a few months to train on some very straightforward, easy measurements that they can be collecting to start measuring soil biodiversity. And then as they're measuring those, we'll work on some of the harder, more difficult trainings to be able to build capacity, to be able to build the infrastructure so that they can eventually work up to where they are doing microbiome sequencing and eDNA sequencing as well. So we want to be able to work up to that, starting at kind of that first level so that everybody is involved, all countries can be involved, and then working our way to building that capacity. So we have now developed these, defined these essential biodiversity variables. And then the next step is where we take this out to the countries. So when the countries that were at the CDB, CBD, apologies, they signed that agreement basically saying we want to measure soil biodiversity in our country, but we need a program to do that. And so what we are hoping is that we will be able to take this back to the countries now and say, we have developed this program, and now we want to give you the opportunity to follow through with that commitment that you made at COP15 to be able to measure soil biodiversity in your country. And so we would work with local stakeholders that are in this international network of soil biodiversity, which would include farmers, researchers, policymakers, and government labs, and we would start to choose those sites. For example, the ones that Manuel pointed out, to say, okay, what are the agriculturally important sites? What are the biodiversity hotspots that we should be looking at? What about urban areas? And be able to kind of develop a plan and say, these are the sites where we should be assessing and monitoring soil biodiversity. And then those countries would then allocate the resources, and then we would be able to work locally to build that infrastructure, training programs, acquire the equipment necessary. And then the next step would be to actually start the monitoring. So you would go out and you would collect the information, and you would basically start to record that in a database that is accessible to all other members, so that I can start comparing my biodiversity with a neighboring country and vice versa. And then kind of the next step is then to say, okay, let's work on advancing to that next tier if we're not already there. And let's start to incorporate these measurements into something that a policymaker can look at and make sense, that they can look at and say, I understand what's going on. And then the second part of that is that we are building up a kind of a list of case studies to be able to say what kind of management practices in agriculture or natural or urban ecosystems preserves and protects and builds soil biodiversity, so that they're able to look and they're able to say, okay, we're concerned about these areas that are starting to lose soil biodiversity. What can we do about that? And then they would have a list of options that they're able to do that they can implement based on the practices that have been reported by others. And so this is what the Global Soil Biodiversity Observatory will look like. I want to point out that we have tried our best to make sure that those essential biodiversity variables that we're looking at are compatible with most of the other initiatives that are already ongoing. So for example, the soil bond is already kind of measuring a lot of these, or the European Lucas project that is already measuring a lot of these biodiversity variables. We want to keep it as much as possible close to that, so that we're not saying, okay, we have to scrap that and start all over again, but that we're saying this is going to be complementary, and a lot of the measurements that are already being collected in that will fit in with this Global Soil Biodiversity Observatory program. But it eventually will rely on the people, on you. And so I would encourage you to join this network. And I'm very excited about the progress that we've made so far, and hoping that we'll be able to take this back to the country soon and get their kind of their endorsement of this. And I believe that is my last slide. And if you do have any questions, please put them in the Q&A, and I would love to discuss those.
Thank you very much, Jacob. Appreciate it. Very, very detailed presentations. Thank you. Probably the last, but probably the most important presentation is from Sandeepa Kanitkar, and she will talk from the industry point of view. Sandeepa.
Thank you. So I hope you can see my screen.
Yes.
Okay. So first of all, thank you, AMI, for this opportunity to share my experiences on the Forum of Soil Biodiversity. Basically, I'm an industrial microbiologist, and we have been trying to use the soil microbiome for industrial applications in areas of soil health, in areas of fertilizer use efficiency, and also in the areas of pest management. So I'll begin my presentation on developing a microbial intensive crop kind of package of practices for many of the issues, because we are targeting some issues which can be solved with the soil microbiome, or you can say phylosphere microbiome. And we have carried out extensive studies at our R&D lab on the high throughput screening aspect and the analysis for the desired activity. Then the microbiome is put through the selection to trial and production for the fermentation, because a microbe may be very good in the community, but when it is subjected to fermentation and formulation, it does not withstand the vagaries which are there in the environment with respect to temperature, with respect to the competition, for access to different nutrients. And then the technology standardization, because as we know, if we have to launch any of the microbes into the system, it has to go through different regulated kind of regulatory bodies. So this is the aspect which is in fact recognized, and we carry out the thorough analysis wherein before the microbe is put into the system, it is put through this rigorous kind of 4 step level processes until it reaches the production stage. So the drivers for using the crop microbiome are, in India, there are a lot of stagnated yields. The issues are there. The farmers are not getting increase for the yields which are they getting right now. They have to keep on using the higher quantity of fertilizers, higher quantity of pesticides, because most of the pesticides, in fact, especially in cotton and rice, they are getting resistance to different pests. So that is a kind of a driver wherein the farmer wants some different solution for whatever he's using right now. The efficiencies which was pointed out earlier, which is carried out by the University of Melbourne, the same in fact is there. When the fertilizer efficiency is extremely less, almost in fact in India, many of the scientists have claimed it is less than 30% or close to 30%. So mainly the farmer is wasting 70% of the nitrogen which is being added, also the phosphorus and the potash. As nitrogen is highly subsidized in India, the farmers tend to use a whole amount of nitrogen, thereby causing greater imbalances in the soil microbiome also and the soils. So this leads to a lot of early kind of degradation. And as the land holding in India is very less with respect to per capita kind of land holding, it is less than 2 hectares. So there, the brunt of this soil fertility reduction is borne by increasing the crop cultivation cost, which in fact burns the farmer, hurts the farmer, because the market prices are, as an American president once said, that farmer buys in retail but sells in wholesale. So as the wholesale prices are not increasing to the extent which the cultivation costs are increasing, there the drivers are there for seeking alternatives to using the chemicals. There is also the problem of residues in the food, because the food safety is going to be very important these days, as India has started exporting right from a food scarce nation in 1960s. It has become a kind of a food surplus nation thanks to the Green Revolution which was there. But right now, when India is trying to export its food produce, especially grapes and pomegranates and different vegetables and horticulture produce, it is facing a lot of problems with respect to the residues which are monitored. So that is the in fact there. The need the safe alternatives, as was mentioned by me earlier. So my in fact company, we are trying to develop a smart bio platform wherein we have access to the biodiversity hotspots which are there in India. And India being a tropical country, we have great, microbiome, in fact, candidates which can be used for commercialization. So the candidates which are used are for the developing different kinds of biostimulants, then the different pesticidal applications, then using these to develop the microbe intensive resilient package of practices, which are because the climate change right now is also hurting. Right now, just yesterday, the New Delhi crossed 52.5 degrees Celsius of temperature, and the farmers, we are bearing the brunt of this increase in temperature. Then the carbon, in fact, in India is the foundation, because in tropical conditions, even if you add a lot of carbon to the soil, that is getting oxidized. So how to maintain and replenish the carbon in the soils is going to be very important. Improving the fertilizer use efficiency with some of the candidates in the soil in the smart bag platform. Then taking these microbes from the lab to the land. That is a great challenge in that, because some candidate, in fact, there is a question over here that what is snake oil? Snake oil is a product which in fact promises the world but doesn't give you the result in the field. So that in fact aspect of microbes giving the plants the property for which it was isolated, that is an application we are standardizing. And we are mostly dealing in the dormant forms of microbes, because if nature has given such kind of dormancy to withstand the vagaries of the temperature, the pH, the salinity, why not use them in formulation? So that is an aspect which we are in fact using in India. So there are different kinds of approaches to developing this microbial bioinoculant. There are and how to find a microbe. So there are some traditional reductive approaches which are there, which can be labor intensive with respect to the which microbe you are trying to isolate based on the carbon, based on the lengthy strain improvement procedures. And then there are limitations also the culturability of the strains, which if you take it in isolation. And the next gen kind of sequencing and the next gen microbials are the one which are not only for selection, but also for fermentation and formulation. So different types of community, in fact, are being assessed by us to find out the best of procedures whereby we could standardize and stabilize the microbiome for its efficiency. So right now we are targeting the targeted screening of plant beneficial microbes with respect to 25 different properties for which we are targeting to isolate this microbiome and then subject them to the various procedures for standardizing them. So the challenges which Brajesh had asked me to mention was the consistency and the quality control of the formulation. That is, in fact, an important aspect. The shelf life and storage of the product which is given, that is an important aspect, and especially for India, where the temperatures go up to 40-45 degrees at the retail counters or at the farmers level. The field performance, initially, a lot of governments, they took a lot of time to standardize and bring out necessary regulatory procedures which are necessary for bringing out a microbial inoculant, maybe for the fertilization or for crop management or for bio kind of stimulant approach or for soil health also.
Sandeepa, can you hear us? I think she got hanged up. Sandeepa? I think
Sorry,
just give her.
Can you hear me,
Sandeepa?
Brajesh?
Yes,
Yeah,
now.
yeah.
Yeah, okay.
Yeah, sorry,
You
there
just
was some.
hanged up for a minute. Thank you.
Yeah, sorry about that. So basically, that is there. Then the regulatory hurdles which are there, we are trying to solve those to a great extent. The economic viability, the compatibility with the existing farming practices, and the public perception which is there right now of these microbes, these are trying to be now solved to a greater extent. The smart bio platform as I was mentioning was with respect to the sample collection. Then how do we in fact isolate the organisms and subject them to the different aspects of temperature, salinity. Then different collaborations are there because this cannot be done by a single company. So we are trying to have the networking aspect wherein we work with different institutes, different colleges in order to improve our microbiome studies and also microbiome applications. I'm just trying to hurry up because I'm having some more slides. So if you have any questions, I would be very happy to answer some of the questions. So as I mentioned earlier, we are trying to find applications of the crop microbiome in different three aspects of crop production cycle. One is the pest management, the nutrient management, and the soil health management. The nutrient management aspect is done with respect to biofertilizers, which we are having basically for for nutrient mobilization. The soil health management is basically used for the carbon sequestration, which is important when you have the residues, for example, like rice, which the farmers burn. So can that be sequestered on the field that is being targeted? And pest and disease management, in fact, is taken with respect to use of biopesticides for targeting specific, in fact, pests with respect to either targeting nematodes or fungi, pathogenic fungi, or some of the insects. So this is being done by a whole battery of microorganisms which we are having. I will just explain the real life examples of the products which we are having, which are being used currently by the farmers in India and also in 5 other countries. So this is the on-farm seed dressing which we are giving the farmers. It's a kind of a dry seed dressing which the farmers can apply because most of the seeds are treated with chemical pesticides and fungicides and insecticides, in fact. And then we are having the compatible microbes which are basically nutrient mobilizing. The farmers can treat them and then they can be used by the field. So it's a kind of a technology which we have developed. We have developed the same technology with the seed companies, wherein the seed companies are using our dormant forms of microbes along with their chemical treatments and the compatibility. In fact, after checking the compatibility of their concoction, then we are targeting and checking the growth of these microbes and stability for almost a year, and then it is produced. So this is right now being used in corn by a multinational company in India, wherein more than 15,000 tons of corn are being treated and they are being used by the Indian farmers with success. So these are the kinds of formulations which are used because we are a thorough microbial company. So you only utilize the benefits of microbiome by selecting different useful microbes and giving it for different applications. So this is the plant health and the abiotic stress management. Someone right now mentioned that the microbiome is an indicator and also a buffer. So the buffering aspect of different microbes are being checked to address the abiotic stresses with respect to drought, temperature, salinity, nutrient, and light. And these kinds of microbes are basically their molecule, they are active ingredients plus the microbes, they are being screened and utilized. Right now, we have developed only 2 products for this biostimulant activity based on microbes only. So these are mainly polymeric kind of substances which are, basically capsule-based ingredients which are being produced under signaling molecules. These are formulated and they are given in the market. This is a unique technology which we have developed to target the rice stubble burning, which is a very problematic issue in India, wherein the capital is engulfed with smog in October and November when the farmers in Punjab, Haryana, they burn their rice stubble. And then they burn because they are not at fault, because the scientists have to give them a solution so that the rice stubble can be put away at a very minimum cost, and the wheat can be sown in 15 days after rice harvesting. So this we have been able to show and standardized, and we have carried out this over more than 60,000 acres in 10 districts of Punjab, Haryana. And this technology right now has won the national award for in situ rice stubble utilization, wherein in 15 days, once we adjust the carbon nitrogen ratio and use it along with the synthetic consortium of this microbes, we can address some of the solutions of this rice stubble burning. So this is a kind of a total kind of market which is there if it is used in the total area of more than 1.5, you can say 10 million acres in Punjab, Haryana. So this is the nutrient use efficiency of some of the microbes which are based on Azotobacter, Bacillus polymixa, and Bacillus licheniformis, which are used in sugarcane, which is a very hungry crop. And there again, the farmer, it is being, in fact, it is considered as a nasty crop for soil fertility because farmers use a lot of water, a lot of irrigation. And then we are trying to improve the fertilizer efficiency dose by reducing the recommended dose of fertilizer and adding the microbials. As you can see over here, we have got around 16% increase in yield over recommended dose. Even if it is reduced to 70%, then we have reported increases. So this is nothing but then increase in the fertilizer use efficiency in a crop like a hungry crop like sugarcane. So this is about the biopesticidal applications wherein we have targeted the use of Bacillus subtilis, Pseudomonas, Trichoderma viride, and Metarhizium anisoplium Bavaria bassiana in different product formulations. Many of these products are formulated as wettable powders. They have been, registered with the Government of India at the Central Insecticide Board. And then we are creating some target examples where, first of all, we check out the compatibility with the routinely used fungicides and chemicals. And that is how the farmers can, be rest assured that even if the tank mix compatibility is assessed, then he will be able to use it intermittently as a part of his IPM procedures. So this is the product which is now being taken up by Bayer in India, our product, and we are also having our product. So it's a non-exclusive kind of a partnership wherein the Rhapsody, the Bayer's product name is Rhapsody. This is used to treat the powdery mildew in grapes, and which is there at the last stages of the grape cycle. So there, in fact, we are getting good control, more than 85 to 90% control if it is used as a part of IPM, and the residue analysis on a 220 residue analysis basis, the farmers have come clean out of it. So this is in fact helping the farmers to mitigate the residues which are there in grapes. The same aspect which is there for pink bollworm in cotton. Now, this is an aspect which troubles the farmers to a great extent. So we have been, in fact, successfully in analyzing the cycles of the insect growth from egg laying to the adult, and we have targeted the egg laying cycles, and 3 to 4 sprays are recommended when the insect lays their eggs, mostly during the new moon day. And this has been, in fact, helping us to solve the problems along with the use of chemical pesticides in an intermittent manner. And more than 70 to 80% of bollworm control were observed in a very difficult area of cotton. In fact, where the farmers are facing a lot of problems with respect to resistance, and some of them, it's very sad that there are many suicide cases also of reducing the yield and increasing cost of cultivation. So we are working with those farmers. So this is how the microbial intensive crop POP works, or package of practices. It looks like, right from seed dressing to the crop residue management. Now this we are trying to take it to the farmers so that they are able to reach the genetic potential and claim the profits in their fields, because it takes a lot of patience and time to develop these POPs. But once the farmers, they understand this, mostly the literate farmers in the horticulture areas, they're able to understand it better, and then they are able to take it further. So now our presence right now is in microbials, because the biological aspect is a huge aspect. We are targeting just the microbiome aspect, the microbial aspect, which is used again in the 4 aspects as I told you earlier. In India, the market right now is increasing because they are trying to find out solutions to the different problems. As I told you, right now the market is around 725 million, and we expect it to increase at a CAGR of more than 15% to almost 1.5 billion. This is a very low estimate, and we expect that it definitely will increase further, because microbiome is used in all 3 aspects of crop production cycle. And the futuristic strategies, I believe, will have a lot of microbials in the system. So that is, in fact, my last slide, and I thank you all. Any questions are welcome. And the presentations before me were very encouraging and motivating, and I would like to address to each of these participants in an individual manner. Thank you so much.
Thank you very much, Sandeep. I appreciate it. I think we still got half an hour to answer a few questions in the Q&A sessions. First of all, can I request each panel member to switch on their camera and video and the but particularly the video, and when you respond, please to make sure that you are unmuted. There is 11 questions in the panel, and then there is other questions that's coming through in the different perspectives. So what I will do, I'll read the questions and maybe specifically ask a particular persons to answer that questions, if that is okay. The first questions was from Anukul Vaishnav. It was addressed directly to me. What do you mean by the genuine and the snake oil? I think Sandeep addressed this issue. There is a lot of product in the market, microbial products there's nothing there. In fact, there was analysis done about the mycorrhizal inoculants that is sold in the worldwide, 75% of those have nothing, no live spore. So if you have a live microbial community, microbial inoculation, that you do not expect that effect on the productivity. So that's what the snake oil is. Just before I started requesting people to answer that questions, can I also request the panel members to keep the response brief? We have a few questions to go through. So maybe answer the question as brief as you can. And if the question isn't answered, and feel free to ask other panel member to respond if you're not comfortable answering that. But we need to answer most of the questions as it come through. The questions from the Brian Dolhany is, what role can the cities play in protecting and harboring soil organism and increasing diversity? Manu, you want to answer that question?
Sure. Yeah. So, I mean, cities, they have a fundamental role, especially in the conservation of organisms, and also in the One Health policies, because we are in touch with our city parks, for example, especially our kids, our children, they are in touch with the city park, and they are training their immune system when they play in the city parks, for example. So cities, they need to really learn more about the microbiomes and how that affect to the human health and the ecosystem health. So they have a fundamental role.
Thank you. Thank you, Manu. Nicola, would you like to answer the questions on what are the key strategy to make soils that can suppress diseases, both in terms of the management tools, but also is there any interventions tools emerging in that area?
Yeah, that's a really nice question. And as we've heard, the microbiota is very location dependent. So the taxonomy is absolutely dependent on where it has been sampled from. So when we're talking about suppressive soils, for example, to suppress some of the fungal plant pathogens, the endemic microbiota might look quite different. The taxonomy might look quite different, but the functions might overlap. So I think one of our main understandings is to look at those functions and to understand, whether they have suppressive and beneficial functions for some of the pathogens. And there's some nice work that has been published in that area. So yeah, I think, as you say, in the name of brevity, try not to go into too much detail, but yes, there has been some good work looking at the functions in particular.
Thank you very much. Jim asked, maybe Ali, if you would like to answer that questions. So Jim asks, microbial inoculation has been used for very long time. That's true from century, honestly. But it has a limited commercial success. How can new approaches changes that? Ali, do you like to answer this?
Sure, that's true. Well, I think that using indigenous microbes has been proven to be a potential way to overcome that issue. And by indigenous or residents, if you want, I mean microbes that come from the area where you want to apply them, or from the plant you want to use them for, because in theory they're better adapted, I suppose, to the environment you're going to introduce them to. So that's one of the approaches, I think, that has been explored the most to increase the success of bioinoculants in the field. Yes.
Thank you, Ali. Hangwei, this is for you, and question is from Mike Dempsey. Can any of the coating you talk about be considered as a plastic? And if so, will they therefore constitute a new microplastic problem?
Okay, thank you, Braj. Before I answer this question, can I also add a little bit for answer for the Jim's question? Yeah, for that microbial soil inoculation with limited commercial success, I think, yeah, we mean limited commercial success. That is compared with current synthetic, for example, the synthetic nitrogen fertilizers. But if you see from Sandeep's presentation, and also in Australia, when I go to the market, I can find many. I would say the biological fertilizers. They have some inoculants with them. They can. I think this is an emerging market. Yeah, although this is currently we have limited commercial success compared with synthetic nitrogen fertilizer, but I think this market is growing, and also the Australian, the biggest fertilizer company are also very interested in the biological fertilizers. And for Mike's question, yes, I think currently commercial available coatings, actually they are, some of them, they have some problems of microplastic, but in our hub, we are trying to develop some new coatings that are biodegradable. That means they will not result in any microplastic or nanoplastic residues in soil. And one of the very promising coatings, what we found is shellac. Shellac is a natural, I would say that's a natural products produced by insects onto the trees. They can be easily degraded and will not cause any negative environmental issues. So that's all from my answer.
Thank you. Thank you, Hangwei. There's a questions that I'm interested in textiles degradations. Have you seen microbiome or ecosystem affected by textiles waste? And how do you think we could combat this? Is anybody wants to tackle this issue? I will do that. Textiles waste have a huge impact on the microbial communities where you put the wastes, because they are really toxic. How do we wants to reduce that? We want to, I guess, use, first of all, there's always to reduce the amount of the chemicals we use in the textile industry, use the more eco-friendly textiles chemicals, and that is more biodegradable. And then the third, before we release in the environment, particularly in the water systems, we must use a processing through the microbial degradations that make sure that the minimum amount of the textile waste goes into the environment, ultimately in the water systems. Another question is, does a healthier soil microbiome add biodegradations? Ellie, you want to tackle that?
Sure, absolutely. Biodegradation is one of the properties of microbiomes, but not all microbes can do that. It's a so-called specialized function of some microbes. So healthy microbiomes and more diverse microbiomes harbor higher chances of having good degraders in them. So, maintaining a healthy microbiome definitely supports biodegradation proper abilities of microorganisms.
Thank you, Ellie. This is a question for you, Sandeepa. It's came from Toby Marique at Plo. I apologize if I pronounce your name wrongly. The question is, nice work, Sandeepa. What are difficulty you facing in implementing the smart bio platform?
Basically, thank you. The implementation of the smart bio platform with respect to different applications has been to stabilize and get the high density fermentations of microbes and then target them to the formulation strategies whereby we would have stable formulations in the market. As you right now mentioned, Han mentioned that he could not find microbes in many of the products. So that is in fact the main character. Microbes work very well in the laboratories, but when you give them to the farmers, they are half as effective, or in fact zero effective at times, because the formulations are not that stabilized. So formulation stabilization is the first key which you have to be able to do it, and then bring out their efficiencies. The strain improvement program has not been used to the extent it has been used in the commercial, pharmaceutical, and other industries, whereby it's a targeted kind of an activity which is taken. But the soil microbiome aspect, in fact, definitely, some of them said that a native biomicrobe, they will function better. But then you will not have a commercial strategy around the native biomicrobe, because you must be looking at how millions of farmers can use a specific product. For example, biopesticides, for example, different kinds of biostimulants. So there, in fact, you have to have a kind of a universal product which can be used by a greater number of farmers, maybe for a specified application. So the main challenges was the formulation technology and the high density fermentations.
Thank you very much, Sandeepa. Jacob, the question is, what does or will GLOSOB do? So I guess the clarification is, will it do monitoring itself, or will it be a repository of data provided by the NetSob members?
Yeah, that's a great question. So GLOSOB right now is basically kind of a scaffolding recommendation of what is to be done. The actual monitoring will be done by the countries following these recommendations. And then, of course, the countries will be able to add their results, the monitoring results, to a central database. And so my ideal would be that members of NetSob, people that the scientific community, would play a critical part in this, in both the monitoring as well as the adding the information to the database, as well as being able to interpret and use the data from GLOSOB to publish papers and to help push policy in both global and national policy.
Thank you. Thank you, Jacob. And Sandeepa, the questions for you, I'll combine them together. From Anukul again, which types of formulations used for the microbial applications under high temperature conditions? And the second question is, Ali, can you please brief about tri-coat technology?
Yes, thank you. So as I mentioned earlier, for the high temperature conditions, we are maintaining the dormant forms of microbes and the mesophilic organisms which are there. So the organisms which are selected also are from the high temperature areas. So I will give you an example of Bacillus subtilis, in fact, which works at a temperature in India up to around 45 to 46 degrees Celsius. And we also have an area in grapes wherein the temperatures during the day are very high, but then during the night they come up to at least 6 or 7 degrees Celsius. So there, in fact, we are using Pseudomonas at the low temperatures, wherein it has got different types of properties like anti-ice nucleation, which can aid it. And the second question about tri-coat technology. Tri-coat technology, we are trying to get the benefits of the nutrient mobilizing microbes and the plant growth promoting microbes with a small kind of a nutrient which is added for the microbes and also for the pest kind of management. So we are developing this tri kind of formulation which can be used because the best way in which you can use the microbes is with the seed. So the better we address the seed treatment technologies, the better it is for the farmers to start at the beginning of his crop production cycle. So I will be able to explain the tri-coat technology if he writes to me one-on-one, and we can then carry on that discussion further. Thank you.
Thank you. Thank you, Sandeepa. Manu, you talked about, this is for you, you talked about that how important the biodiversity is and an important role they play in the productivity. But what are the approaches that is available today to manipulate the soil biodiversity in agroecosystem?
In an ecosystem, like the most traditional way to change the biodiversity is through amendments, organic amendments. So pretty much, and changing the pH of the soil. Although most of them they are indirect. So we have been adding organic matter to the soil for centuries. We have been changing the pH of our soil, adding calcium to our soil. That is having an indirect influence on the biodiversity of the soil, and that was not intentional. Anyway, and these days, I guess that the most direct way to influence the biodiversity of the soil is through the probiotics. But as you guys are mentioning in this discussion, many of them, they are not really working because they are not able to colonize the soil and establish in the soil. So there is like a huge challenge there, and many opportunities for changing the biodiversity in the soil. One of them is what I was talking in the presentation, the native microbiome, to try to fuel certain species that they are already in the microbiome, and then they can play an important role in the productivity of the plants. So probably that's a way to move this forward.
Thank you very much, Manu. The one more questions for Sandeepa. Sandeepa, are these bioinoculants compatible with GMOs? Any experience with relative efficacy? I don't know what the second questions mean about relative efficacy against what, but I guess the first question is that can your bioinoculants be used along with the GMOs? I'm guessing that is particularly relevant for the cotton industry you were talking about, Sandeepa.
Yeah, GMOs will be the genetic engineered microorganisms, not I think so BT cotton. But in India, we are yet not following the we cannot release the genetically engineered microorganisms into the environment. But with respect to BT cotton, definitely we have got good results with respect to the BT cotton, because most of the cotton in India right now, more than 85-90% of the cotton is BT cotton. So yes, we have got very good results in BT cotton also. With the GMOs, we have not yet tried it because in India it is not allowed. Some of our products, farmers which are doing that in US, I'm not aware of that. Sorry, so I can't help you with those.
Thank you, Sandeepa. The questions for you, Hang Wei. I think that is a follow-up questions about your microplastic answer. This is again from the Mike Tempsey. He asked that I recently read that shellac was replaced with the plastic about 100 years ago because its production is limited. Therefore, won't that limitation still apply?
Yes, thank you, Mike. I'm not sure whether I can answer your question very well because this project is conducted by our collaborators who are chemical engineering scientists. But from what I can understand, shellac currently probably it's not its production is not very limited because I learned from their presentation shellac is widely used in some food industry. And but I'm not sure my memory serves me right or not. And in the supermarket, and if you go to the supermarket, some of the foods actually that use the shellac. Shellac can be eaten by humans, and so that's why I think current days shellac production has no limitations. Yeah, that's my understanding.
Thank you, Han Wei, appreciate it. Ali, this is for you. The restorations industry has been one of the industries which has been reluctant in use of the microbial inoculants. The focus has been so much on the plantations. How can we change this going forward?
Well, good question. I think that there are many gaps in the knowledge at the moment on what are the requirements of these plants, native plants, for example, in a restoration, and what are the mutualistic interactions that need to be reestablished? Because unlike crop productions, where are monocultures usually, right? In natural environments, you have many plants and very complex communities. And this is one of the main challenges that need to be overcome. So I think that deeper understanding on the nature of these interactions is needed. So more research in there. And also, I think for restorators or restoration practitioners to be open to this, looking below ground and looking at these interactions. Most of them are. So more science around that, in incorporating that. We are lacking behind, I think, compared to crop and agriculture in that sense. Yeah.
Thank you. Thank you, Ellie. I guess this question is for Nicola. Nicola, you talked about the cryopreservations. The question is that how many samples is physically and logistically possible, soil samples, soil microbiomes, so they're saving soil microbiomes, so you're preserving the whole soil. Guess what is the target in that projects or the national targets, that how much soil or how many soil samples can be cryopreserved for the future uses?
Yeah, again, it's a very good question. So for the culturable microbes, so microbial isolates, that was quite straightforward. And those are obviously the long-term indefinite biobanks that we have created. So we have about 30,000 isolates, I think about 32,000 isolates at the moment. But for the microbiomes, or the microbiota, I should say, that was very interesting. So for this project, we were looking at the rhizosphere soil, so the thin layer of soil associated with the root. So that was removed from the root. And our partners in CABI have developed a cryopreservation technique where they can make small beads of particles containing this material. So this is not cultured, it is just processed and then stored directly in liquid nitrogen. Now, there'll be a finite number of beads that are preserved, unlike the microbial isolates, which can be preserved indefinitely. So, it's a very interesting technology. They have tried to maximize the amount of beads that they can store, and each can be stored, several beads can be stored in a single cryovial, and it's just housed under liquid nitrogen. So the main limitations are A, the amount of starting material, and B, the costs associated with liquid nitrogen storage, and of course the space as well. But it's an interesting development. Sorry.
Thank you. Yes, it definitely is. I think the last question is for Jacob. Jacob, you talk about the 3 different tiers of measurements or monitoring, and you also talk this is because of the logistic problem, and that's a logistic, I guess, in infrastructure inequity. Can you just give one example? What do you mean by that logistic problems, and that's required to have these 3 tiers of measurements and monitoring?
Yeah, sure, absolutely. So the idea is that there are some countries that have almost no infrastructure to be able to do biodiversity measurements that we would like. So for example, this webinar is about microbiomes. There are some countries that don't have the people, the personnel that are trained to be able to do, let alone sequencing capabilities. And so we can't just come in there and say, you need to start sequencing. So instead, in those cases, we're going to go in and we're going to say, well, let's start looking at enzyme activities in soils, and let's start measuring some of the macrofauna that are in the soils that you can identify. And so we'll start there, and then we'll work with those countries to be able to build those infrastructures, to be able to work and say, let's get you a sequencer. Or, if you don't want a sequencer because this is all up to the countries, then maybe there's a regional laboratory that can be responsible for kind of sequencing for several different countries, just to be able to kind of put those logistics together.
Thank you. Thank you very much, Jacob. I think we have run out of the time. So I'll start with thanking our panel member for taking time and for excellent presentations and question hour sessions. AMI has done a wonderful job to get everybody here and organize everything. So thank you very much, AMI. But last, but probably the most importantly, all the participants, you have made this webinars and panel discussions really lively and useful. And the questions, I'm also, and this is a kudos to all the panel members and AMI. There's a lot of message of very insightful talks and discussions. So I will take this opportunity to thank you, all of you, and thank you all our participants, both the panel member, but also all the people who participated and attended as an audience and asked those questions. That has been wonderful time. So thank you very much, everybody. Thank you very much, and have a good evening and a good day
Thank
where
you.
you are depending upon that.
Thank you, Braj.
Thank you, Raj.
Thank
Take
you.
care.
Thank you.
Thank you.